Welcome to Flare Inc.'s animated course content demo reel. In this video, we will learn about DNA microarray. It comes under the course Molecular Diagnostics. Let's say you're an enthusiastic researcher who's about to work on a gene expression experiment. You're placed in a well-equipped laboratory and you're being provided with a bunch of cDNA strands, whose potential expressions you're going to study in your sample of interest. So, how do we perform this task? We start by preparing our sample of interest. Let's say you want to study the difference in gene expression between a normal cell and a cancerous cell. You prepare complementary DNA strands out of their genetic material. You label these cDNA strands using fluorescent compounds to identify their origin. Once your sample preparation is done, you fetch a gene, the expression of which you want to study in your sample, and you place them along with your sample to check if they hybridize. The hybridization occurs only if your sample expresses this particular gene. The sample which does not hybridize gets removed from our mixture. Using standard detection technique, we check which of our sample has expressed the gene of interest. Since our samples are fluorescent labeled, they glow when light of appropriate wavelength interacts with them. If our sample responds to red light alone, it means our cancer cell has expressed this particular gene. If our sample responds to green light alone, it means our normal cell has expressed this gene of interest. If our sample responds to both red and green light, it simply means that this gene is expressed in both our cancerous and normal cells. You have successfully studied the expression of a single gene among your sample. There are thousands of other genes whose expression has to be studied for the same sample. This leaves us with a large number of cDNA strands whose expressions we have to identify in our sample. So how can we perform analysis on all these samples in an efficient way? We can hire a bunch of talented people to perform this task, or we can create a machine that automates this routine for all our cDNA strands. But both of these approaches have a serious downside. The amount of time demanded for processing all these samples is way too long, and the amount of money spent on chemicals and talented workforce is too high to be justified. So how can we address these challenges without compromising the quality and the accuracy of your experiment? This is where microarray comes into play. DNA microarray helps you to perform multiple gene expression studies simultaneously, all within an area as small as a microscopic slide. Let's zoom in on this slide to have a clear idea. Each DNA microarray holds a large number of segments. Each of these segments holds a bunch of cDNA strands that are placed with machine precision. Each of these strands are uniquely identifiable and are immobilized on the surface of our microarray. Each of the cDNA strand will help us to identify a specific defect in our sample. For example, one of these strands, if expressed, indicates that your sample has cancer-causing gene in them, and the expression of another strand might indicate that your sample has sickle cell anemia-causing gene in them. This helps you to speed up your gene expression study while saving your time as well as a lot of resources. For simplifying our visualization, let's replace each segment with just a single cDNA strand. A solution containing our samples is allowed to interact with our microarray. Our sample then hybridizes with the cDNA strands. The rest of our sample will be washed off. Now we proceed to identify which of these genes are expressed in our sample. We scan our microarray using a green laser. The illuminated spot represents the genes that are expressed in our normal cell. We then scan again using a red laser. This time, all the illuminated spots corresponds to the genes that are expressed in our cancerous cell. We fetch both these separate results and we combine them to form our final output. Now you might be fascinated to find an orange spot in our final result. It simply points towards a gene that is expressed in both our normal cell and our cancerous cell. Remember, the color in our microarray result corresponds to the sample in which the gene has expressed itself. Since each strand in our microarray is uniquely identifiable, we can understand how normal cell differs from cancer cell in terms of gene expression just by reading the output. This helps you to save a lot of time and resource without compromising the extent of your experiment. This is the beauty of microarray. Hope you guys found this video helpful. Thank you for watching.